statements contained in this presentation have not been evaluated by the Food and Drug Administration. The products discussed are not intended to diagnose, treat, cure, or prevent any disease.
Okay, good evening, America, and good morning, Philippines, and the rest of the world. So the role of epigenetics in gene expression, in the context of gene expression, and it is now being used as one of the medical intervention therapeutics or modern medicine, which is actually geared towards fixing what you call the genetic problems, as well as fixing some physiological due to genetic disorders or genetic fallout or disarray. So the role of epigenetics in gene expression. So before we could really go deeper into the what is epigenetic medicine and how is it being carried out, we will now review our concepts of basic genetics. Here are some basic concepts in genetics. Take note, basic concepts in genetics. So genetic information. A gene is defined as the basic unit of genetic information that determines the inherited characteristics. Every characteristic or every trait that you displayed in your body is actually underlined by a particular unit called gene. So every protein is actually a gene in a one gene, one protein theory. So if you have about 100 or 30,000 proteins, we have also 30,000 genes because one gene expresses a genetic trait which is actually the protein which is shown in your characteristic. The color of your hair, the color of your eyes, the color of your skin, your intelligence, every trait that you have in your person is actually due to that material called gene. It is the basic unit of genetic information that codes for a characteristic that you have seen in your person. The collections of this genetic information is called genome. The whole collection of genetic recipe is called genome. And that genome is actually housed in the chromosomal structure called chromosomes. Chromosomes, therefore, is a conglomerate of genetic genes, material, DNA plus proteins organized into 46 chromosomes in the human body. So it is the storage units of all the genes. So you can see there in the diagram, the tetrad, two long arms and two short arms connected through a centromere. You call it tetrad, and you see it is, and that particular tetrad is actually due to the double helix structure, which is the baseline or the basic structure unit of genes. You see there that genes are then embedded in that double helix structure as described by Watson and Crick, no? That particular DNA double helix is a nucleic acid that contains the genetic instructions or the recipe specifying the biological development of all cellular forms of life. Every protein in your body is actually due to that particle DNA, and that DNA group into a certain sequence that codes for a protein is called the gene. So the gene is also DNA, but the gene is simply the DNA with a equivalent protein. There are DNA sequences that has no equivalent protein, and we call that introns. We call that the nonsense codes. So there are DNA sequences in our chromosome which has no protein equivalent, no recipe at all. They are just blank. These are called introns. And we have 90% of that in our DNA structure. So if you are going to describe it, DNA are just simply the polymers or the chain of nucleotide bases that forms into a DNA structure and is the basic unit structure, and it's called double helix. So you have this packaging of the DNA. So on the top, you have the basic fundamental structure, which is a 2 nanometer fiber, which is double helix. And this fiber of DNA or the chain of DNA is like on a beads of a string on a what you call histone protein. So when the DNA is then started now to be wound up on a string of histone, it is called beads on a string of, so those genes are then now bound to a histone. Remember my lecture last time, the histone is a protein that holds the gene. When the DNA is wound up into that histone, it becomes now an 11 nanometer fiber. And then that 11 nanometer fiber is then further folded into to become a 30 nanometer chromatin fiber of packaged nucleosomes. The nucleus or the histones are packed together. Histone 1, histone 2A, histone 2B, histone 3A, and histone H4, H3, and H4 becomes now a nucleosome. So nucleosomes are composed of 4 histones that is now packaged in what you call the 30 nanometer fiber. So imagine that 30 nanometer fiber is folded in another domain and it becomes a 300 nanometer fiber. And that 300 nanometer fiber is further folded to become a 700 nanometer fiber. And that 700 nanometer fiber is then organized into a tetrad. So the one that you can see now in the chromosome, 2 short arms and 2 long arms are then the one that you can see during mitotic division, particularly during the metaphase stage of the cell division. So the chromosome is very visible during the metaphase stage of cell division. So if you are going to look at it, if you unwind that tetrad, it becomes a 2 nanometer fiber, which is the basic fundamental structure of the DNA. So the DNA at the molecular level is naked. The DNA without the histones is naked. With the histones, it is now organized further into a thicker fiber, 11, and that thicker fiber is folded into another folding. It becomes 30, and that 30 is folded in another folding. It becomes 300, and that 300 is folded further. It becomes 700, and that 700 is folded, and it becomes a tetrad. It is the chromosome now you can see is 1,400 nanometers, and that is visible already in the microscope. And one chromosome, as I told you before, is about 150 nucleotides or more. If you are going to put one DNA to another DNA or one nucleotide to another nucleotide connected in a helix, every million letters of the nucleotides takes a linear distance of 0.34 centimeter. And therefore, if you connect all those and all those nucleotides in a chromosome, say 150 million nucleotides in one chromosome, multiply it with 46 chromosome and multiply it with 300 trillion cells. And in every cell, there are 46 chromosomes. So multiply it with that 150 million. It will reach the moon and back to Earth 3,000 times. That's the length of our human chromosome. The chromosome logical structure. So the locus, there is a term that we introduced here. The locus is the actual location of a gene marker on the chromosome. So the locus identify the location of the gene in a chromosome. So there is a pair of gene in every chromosome. So take note, there is a pair of gene in every chromosome because one member of the pair comes from the mother, one member of the pair comes from the father. That's why all genes occur in pairs and we call it allele. I repeat, allele. So in one locus, for example, you may have 2 alleles. Say one locus marker has 2 alleles, allele 1 and allele 2. Or another locus may have possible 3 alleles, allele B1, B2, and B3. So allele is a pair of gene. So take note that characteristics are governed by a pair of gene or pair or more. Your characteristic intelligence may be because of so many alleles. the more alleles you have, the more intelligent you are. So, say the more alleles in that member of the locus of a particular gene trait, then the better is the expression. So take note, there are several alleles. There are several pair of genes in one locus. So human genome, most human contains, of course, the human chromosome contains 46 pairs of chromosomes, 2 sex chromosomes, X and Y, and there are 22 pairs of chromosomes called autosomal gene or autosomal chromosomes. So our chromosomes are divided into two groups. The 22 pairs or the 44 is what we call autosomes. And the second last pair is called the sex chromosome. It is a pair of X and Y. XX if it is female, XY in males. So these are what you call sex chromosome and autosomes. So our chromosomes are organized into 46 chromosomes from the largest to the smallest. So you will notice there, the arrangement of the chromosome is from the largest. So chromosome 1 is the largest, then followed by chromosome 2, 3, 4 down the line up to the X and Y pair or the sex chromosome. The concept of genotypes versus that of phenotypes. Each locus, as I told you earlier, and if the color is brown, then that is what you call the phenotype. And if the color is brown, then that is what you call the phenotype. This constitutes what you call the genotype. So for example, the color of your hair is governed by a pair of gene, which is called the genotype. And if the color is brown, then that is what you call the phenotype. So the composition of the gene where the gene is made of is called genotype, whereas the expression of that gene that you can see or visible is what you call phenotype. Therefore, the expression of a genotype is known as phenotype. So when you're asked, what is your phenotype? Your phenotype is curly hair. That is a phenotype. What is your phenotype? White or fair skin is your phenotype. Or what is your phenotype? Straight hair. Or the other person has a curly hair or a kinky hair. So the expression is what you call phenotype. The one you see is the phenotype, but inside that particular trait is what you call the genotype. The hair, the color of the hair, the weight, or the presence or absence of a disease is what you call the phenotype. So once already seen or once expressed, it is already what you call phenotype. But the genes comprising that particular phenotype is called genotype. So let us have some review of the genetics, no? So the DNA is a sequence of nucleotides, and once the sequence has equivalent protein, you call it gene. But if the DNA has no equivalent protein, it's called intron. So a gene is a sequence of DNA with equivalent protein, whereas a DNA sequence with no equivalent protein is called intron or nonsense codons. So let's try to look at it. The DNA is the gene or the recipe of that protein. The DNA is a process called transcription. So the DNA is transcribed into RNA molecule because the DNA is not movable. So it has to be transcribed first to RNA because DNA is not movable. Only RNA can travel from one point of the cell to another. So that process is known as transcription. But remember that the DNA has nonsense codes, or the DNA has the genes must be edited before it becomes a final copy of the transcript in order to translate that into protein. So what they call the process is known as splicing. You call it RNA processing or splicing. So what is the purpose of splicing? Primarily to remove the introns before they become the final or mature messenger RNA. So before they become a messenger, all those intervening sequence, the nonsense sequence, must be removed in the process called RNA processing. So RNA processing is part of the genetic processing to remove the introns or to remove the intervening sequence which are actually placed by God there to protect us from possible damage. So once the removal of the introns are done, it will now become what we call the mature messenger RNA or the exons. So that messenger RNA is then translated into proteins. But translation involves a lot of modifications, especially that proteins are simply the sequence of amino acids dictated by the sequence of the nucleotides. So the sequence of nucleotides will dictate the sequence of amino acids. The sequence of amino acid is translated into protein, but sequence of amino acids has no meaning until they are folded into a particular structure that will now configure them into a functional protein. So meaning to say that what I'm saying is a protein is not functional until they are folded into a certain configuration. Then you probably ask me, Doctor, what determines their kind of folding? So what determines their kind of structure? The kind of structure unfolding is determined by the sequence of amino acids. So the sequence again is the culprit for the folding, the structure. Kung anong itsura ng folding niya, depende yan. It depends now on the sequence of the amino acids. And take note that the sequence of amino acids is dictated by the recipe of the DNA. So the DNA dictates the sequence of nucleotides. The nucleotides dictates the sequence of the messenger RNA. And that messenger RNA dictates the sequence of amino acids. And the sequence of amino acids determine the folding of that protein until it becomes a functional protein. So until the modifications are made, the folding, the additions of some sugar, and it now becomes a functional protein. But this functional protein comes in a vicious cycle because protein has to be degraded in order to provide the amino acids. Remember that the proteins are sequence of amino acids. So the gene will just dictate what kind of sequence it is, but the cell will get that sequence from its own protein. So meaning to say, we have to provide ourselves with amino acids in order that the sequence will be translated into a functional protein. So one mechanism is that the protein must be degraded in what you call proteolysis. So through the proteolysis will now provide amino acids. Peptides and amino acids are the products of the degradation, or proteins are digested in the process called proteolysis. The proteolysis will now provide our cells with peptides and amino acids. So when we eat protein, they are digested and they undergo what you call the proteolysis, so that the proteins that we eat will provide the raw materials, the peptides and amino acids for the reinforcement of the translation of protein. Take note, there is no translation of protein from the recipe of the gene without the available amino acids. So probably you ask, Doctor, is that amino acid provided only by food? No. Can also be provided by the proteins inside the cell, especially if you don't eat. If you don't eat, our body will utilize your own protein. It will be degraded to provide amino acids in order to translate that recipe into a protein. So it is actually a self-supporting mechanism inside our cell, but that is very bad because if you subsist only on your own protein without additional input of new proteins, then time will come when your protein will all be consumed and you will die. Therefore, in order for us to survive, we have to supply ourselves with the raw materials of protein so that when this protein are digested, they will provide amino acids and peptides in order for the recipe to be expressed. Remember, the DNA or the recipe cannot be translated into protein without the available raw materials such as the amino acids and peptides and oligopeptides. At this point in time, we will now inject the idea that our Imurigen total complete supplementation, because it supplies not only the recipe, but it supplies also the oligopeptides, the 17 amino acids that is derived bovine sources. Remember that the bovine sources are sources of amino acids too. There are 17 out of 20. And it is called complete protein because in that 17 amino acids, all the essential amino acids for protein translation is present. Any protein that has the essential amino acid is considered to be a complete protein. So our Imurigen is a complete supplementation because it has 17. And out of that 17, there exists the 9 essential amino acids that the body needs for protein synthesis. All the other amino acids are provided for in our diet. So don't worry, if you just continue eating, you will have your amino acids. And then if you supplement Imurigen, you will be provided with the essential amino acids because essential amino acids has to be supplied because these amino acids cannot be synthesized by our body. Ordinarily, our body can synthesize amino acids from sugar. So by just eating sugar, if you eat sugar or carbohydrates, your body provides 11 amino acids. That 11 amino acids plus the essential amino acids will now become complete amino acid to provide yourself for the protein synthesis to translate the DNA as the recipe into the actual protein, which is actually the expression of the gene. So if you may ask right now, all proteins in the body are the phenotypes, or all proteins of the body are called the expression of the recipe. I repeat, all proteins in our body are the expression of that recipe in the gene. So take note, that recipe in the gene cannot be expressed into protein without the additional amino acids from our food. So we have to eat our proteins in the diet so that we will be provided with the raw materials. That is the flow of the central dogma of life on which life is anchored to. So life is anchored in this very fundamental structure. There is a change in the gene, there is a change in the protein. If there is a change in the protein, there is a change in structure, there is a change in function. That's how we get sick. If that function is changed, then we get sick. I repeat, when there is a change in that function, when there is a change in that structure, because of the change in the sequence, it will now cause diseases. So that is what you call degenerative and genetic diseases due to the altered recipe. So you see, the peptides and amino acids coming from the protein that our body manufactures will reinforce the process called translation. Because the process called translation in the central dogma of life is to make that messenger into the actual product, which is the protein. So the protein, which is the product, reinforces the translation process in the genetic processes in the central dogma of life. So this is the caricature of the flow of information. So inside the nucleus of our cells is the DNA, and that DNA is duplicated to sustain the living cell. So remember that the living cell cannot be sustained without replication. And every expression of that recipe requires what you call the process called transcription, wherein the DNA is then converted into RNA. You call it the messenger RNA. Only then, when the messenger RNA is completed after editing, it is transported to the cytoplasm, particularly in the ribosome, where will now translate the code into protein, and proteins are then distributed throughout the parts of the body to make the function. That explains now the caricature. Here is another caricature or illustration where a certain segment of the DNA double helix is being transcribed The one you see that the one in red is the transcribed structure. It is unwound. So the double helix is unwound and is unwound under. Then it is being read as the template, and the messenger RNA is transcribed from there through the enzyme called RNA polymerase. Take note that these RNA polymerase are enzymes. And I can surmise that in our immunogen battle, there are RNA polymerases that are present there because our immunogen battle, according to the scientists who visited the Philippines last year, last June, they said that one of the functions of immunogen is for gene expression or transcription. So in other words, they are saying that our immunogen battle has an enzyme called RNA polymerase to provide our cells with the messenger RNA. Again, messenger RNA. So our immunogen is capable of inducing the cells in our body to produce what you call the messenger RNA that our cells will require. So that is what you call the RNA template, which is the recipe. The recipe is coming from the DNA, and that recipe is transcribed into RNA, and that RNA is transported to the ribosome for the final making of the proteins in that protein producing machine, which is the ribosome. So this is the structure of ribosome. The one you see in orange is what you call the ribosome. So the one you see, the line there, the codons are actually transcribed recipe from the DNA. And the codon exists in triple nucleotides. So GGG, that's the first. They have the AAA, then it's followed by UCG, then it's followed by GUC. And every codon has what you call the anticodon. The anticodon is actually the sequence that will recognize the codon, and that is actually the role of what you call the transfer RNA. So our body has 20 different amino acids. There are also 20 different types of transfer RNA because only one type of transfer RNA can carry an amino acid. So the amino acids from the food we eat is then carried by what you call the transfer RNA, and that transfer RNA will pick up that amino acid from the cytoplasm and bring it to the ribosome as dictated by the recipe of the codon, the transcribed codon. So ma-imagine mo that the recipe is then converted already into a messenger, and the messenger attaches to the ribosome, and the ribosome is then being read by the transfer RNA, and specific amino acids come into the site according to the anticodon present in the transfer RNA. So take note, only one specific amino acid is being picked up by specific transfer RNA. So since there are 20 different amino acids, there are also 20 different types of transfer RNA. So it's the transfer RNA that picks up the available amino acids from the food we eat. That amino acid is being picked up and it is brought to the ribosome so that it will now be connected to each other in what we call the polypeptide chain. Because an amino acid, one amino acid is connected to another amino acid by a bond called peptide. That's why the chain of amino acid is called polypeptides. That's why you have polypeptides, oligopeptides, polypeptides, or oligopeptides. Peptides are chain of amino acid, which is dictated by the transfer RNA or pick up by transfer RNA to the ribosome because the ribosome has the codon. So this is the example of the codon. So the first letter, because there are 3 letters in a codon, the first letter, the second letter, and the third letter. So there we have example of a codon. U is phenylalanine. UC is another phenylalanine. Then UUA is leucine, and UUG is leucine. CUU is leucine. CUC is leucine. So these are what you call codons. So this is what I told you before, that our codes, our amino acids are redundant, meaning to say there are more than one codon in one amino acid. So in the case of leucine, for example, as you can see in that drawing in this table, there are 6 codons in leucine. You have the UUA is leucine, UUG is leucine, CUU is leucine, CUC is leucine, CUA is leucine, and CUG is leucine. Now, question. What is the importance of the redundant code in our body? The importance of these redundant codes in our body is to prevent mutation. Because earlier in my discussion before, if there is a change in one single amino acid, it might be possible to have a change in amino acid. And change in amino acid will go to a change in structure. And change in structure will change the function. And change in function will now cause disease. But because of the redundant codes, it will prevent the mutation, because if, for example, A is replaced by C or C is replaced by G, but the same second and first letter, UUA and UUG is still leucine. So even if there was a change in nucleotide, there was no change in amino acid, and therefore no mutation because of the redundant codes. That's why if you are going to ask me, what is the importance of the redundant codes? The importance of the redundant codes is to prevent mutation to occur in our body. Let us thank the Lord for that redundant codons. For example, in this table, please look at arginine or ARG. Arginine has how many codons? There are 4. CGU, CGC, CGA, and CGG. So if your CGU is changed to CGC, there is no mutation because CGU is arginine and mutated to CGC, which is also arginine. So in other words, there is no change in sequence. So if there is no change in sequence, there is no change in structure. So therefore, no mutation. That is the importance of the redundant codes. Remember that our body has their own repair mechanism. If they detect a certain error, they will replace it with the correct one, and then eventually it will make the same amino acid, and therefore it will prevent mutation. Imurgine extracts in that bottle contains those enzymes that will ensure the redundant codes to be available in our body in order to prevent mutation. That's why Imurgine is very notorious as a supplement to prevent mutation because of these available redundant codes. So the redundant codes are useful in our body. There are also promoters and enhancers or silencers in the gene. Remember, the gene expression is nothing without the promoter. The recipe cannot be translated into protein without what you call the promoters, enhancers, and silencers. So you can see there in this diagram here, there are proteins that are what you call activators. As I told you, there is an RNA polymerase. Our Imurgine bottle contains RNA polymerase. And remember, the RNA polymerase is an enzyme that will now translate the recipe of the gene into an RNA. So take note, our Imurgine bottle has an RNA polymerase. Added from the RNA polymerase, you have the proteins called the TATA binding proteins. You have protein 110, protein number 80, all those number that you can see there, 250, 40, 60, 30 beta alpha, 50 beta, the activators, one activator to another activator. There are silencers, there are repressors. These are what you call the epigenetic proteins. So here is now the concept of epigenetics. The DNA is the genetics. The operator of that genetics are what you call epigenetics. So if you ask me, where can you find the activators and the enhancers? You can find the activators in the nucleoplasm of the cell. So in other words, protein that will bind to the DNA are what you call promoters and enhancers or inducers. They can speed up what you call the transcription, because transcription is the act of making the gene active. I repeat, the transcription of the recipe from DNA to RNA is what you call gene induction or gene expression. So therefore, at this point in time, there is no expression of the recipe without the epigenetic factors. The one that you consider, the activators, the silencers, the repressors, the promoters, and the RNA polymerases are what you call the epigenetic proteins. These are the epigenes that are found in the cytoplasm or in the nucleoplasm of our cells. So if you ask me, where can you get these enhancers? We can get it from the food we eat. That is why Imurigine is a very providential supplement that is given by God to us, because the Imurigine has provided us all these activators, all these enhancers that are present in the Imurigine bottle. So the extracts coming from the bovine sources are the sources of what we call the promoters, the enhancers, and silencers. Then you probably ask, Doctor, what if I did not take Imurigine? You can still get it from the food you eat. That's why we have to eat every day. And because the food we eat every day is not complete, we really have to supplement Imurigine as our providers of enhancers and providers of some repressors or silencers of the gene that operates the genetic operation inside the cell. So now you can see in this caricature, DNA is transcribed to RNA, then RNA removes the introns for processing, and then we have the mature RNA, and the mature RNA is transported into the ribosome for translating into the actual amino acid sequence or the protein byproduct. So the question is, how many genes do we have in the human body? It was estimated that there are about 30,000 genes in the human body. Therefore, there are about 30,000 proteins in our body. But the answer to this question is always meaningless, because you see, the effect of epigenetic factors or the enhancers could alter the sequence of the amino acids by splicing. Therefore, altering the sequence by splicing is one way of making another protein. Therefore, the answer to this question, even if we have 30,000 genes, we have more than that, because even if we have 30,000 genes, our body can make new proteins to protect itself. For example, you lack water. Our body now will now provide protein that will now preserve water. That is the meaning of being able to cope up with the. So the epigenetic factors are possible regulators to make new proteins for the survival of mankind. So the answer to this question of how many genes do we have, although basically we have only 30,000 genes, it could be more than that. Why? Because each gene can give rise to several proteins by what you call alternative splicing. Remember that the mature RNA is a product. Therefore, alternative splicing could give rise to different sequence, and different sequence could give rise to another protein, which could be a source of protection for our human body. So epigenetic, therefore, epigenetic therapy, for example, epigenetic medicine is anchored on the alternative splicing. That is why one form of epigenetic medicine is supplementation of vitamins and minerals, or supplementations of nucleotides, or supplementations of oligopeptides is considered to be an epigenetic medicine therapy. If you are going to ask me what is epigenetic medicine, epigenetic medicine is actually a medicine of supplementation, supplementing nucleotides, supplementing oligopeptides, supplementing minerals to operate the gene or the recipe to be expressed is what we call as epigenetic medicine. So realizing that genes cannot be expressed without epigenes, then the epigenes or the supplements are very crucial in producing new proteins to protect ourselves, or producing proteins that will silence the bad genes. To silence the bad genes requires also alternative splicing. So if you want to be protected from some sort of a disease, then we need to have what you call alternative splicing. So alternative splicing is one form of what you call epigenetic medicine. Peptide therapy, nucleotide therapy is one form of making an alternative splicing to make new proteins for a new function to protect our survival and to protect or prolong the lives of the person. So for us to be able to survive in an extreme condition, we need to have alternative splicing. So the answer of how many genes do we have is almost meaningless because each gene that resides in the chromosome can give rise to several proteins by what you call alternative splicing. And alternative splicing is a function of epigenetic proteins. Each protein can be modified in a multiple ways by one is phosphorylation. Phosphorylation is almost equated with activation. So when one protein or when one molecule is phosphorylated, it is activated. Methylation is, as mentioned by Dr. Arlie, that I will discuss about methylation. Methyl group is actually carbon with 3 hydrogen. You call it methyl group. Methylation is almost equated with silencing of the gene. If you want your gene to be silenced or the bad genes to be silent, then it has to be methylated. The addition of methyl group requires an enzyme that comes from the cytoplasmic proteins, and methylation is a function of the epigenetic inductors. In my assumption, our imorigin bottle has a particular enzyme that is capable of methylation, or our imorigin bottle has a particular enzyme that would allow methylation because methylation is silencing the bad genes. So when our body encounters a bad gene, then it's time for methylation. So methylation is one modification to silence the gene. Phosphorylation is equated with activation. Methylation is equated with silencing the gene. Acetylation is activating the gene because acetylation is equated with the unpacking of the chromosome. We say that the gene is held in a histone. So acetylation will allow the duplication of the DNA because acetylation will remove the histone from the gene so that the DNA can be translated or the DNA can be transcribed. So that is acetylation. Glycosylation is another enzyme or mechanism, addition of sugar in order to modify our proteins. Many of the proteins that are found in the membranes of our cells are glycated. So they are called glycoproteins. Many receptors of hormones, many channel proteins of our body, the channel for sodium, the channel for amino acid, the channel for glucose are made of glycoproteins. And glycoproteins is a product of glycosylation in the endoplasmic reticulum of our cells. I repeat, in other words, glycosylation is present in our cells. And I can surmise that our imorigin bottle contains enzymes that will allow acetylation because acetylation favors what you call DNA transcription. Acetylation favors DNA translation and DNA transcription. Phosphorylation also favors transcription. Methylation favors silencing of the gene. So these are the functions of the epigenetic factors. These modified proteins can then further take part in the different protein complexes to modify the functions in case the person is suffering from emergency or circumstances that are very new. If we encounter some circumstances like we are being attacked by viruses, our protein complexes are then modified in order to produce antibodies. So modification, like production of antibodies, is one form of modification of proteins that will allow our survival. So, and this modification is due to either phosphorylation, methylation, acetylation, glycosylation, on which enzymes are present in our imorigin bottle. What is the epigenetics? The epigenetics is the study of the processes by which the gene will give rise to another phenotype. The phenotype is changed in order for us to survive. The changes in gene activity during the development are actually underscored by epigenetic factors. So when we are still in the womb or mother and we develop our parts of our body, that development is actually a function of the epigenetic factors. So the development of the fetus inside the womb of the mother is actually a typical example of epigenetic mechanisms in order to develop the parts of our body. So the development of our organs is actually an epigenetic function. The development of the parts of our body is a function of epigenetic factors. The power of epigenetics is that epigenetic defines the processes by which the organisms can access and use the information stored in the DNA in order to be modified and be able to us to survive. Epigenetics, therefore, can tell us how do we interact with our environment. So as at what I mentioned earlier, supposing you are in a desert and there is no water, our body will now provide new proteins to protect us from desiccation so that water can be conserved in our body. That is one form of epigenetic mechanism for us to allow to survive. We escape the tyranny of DNA. So we escape the tyranny of the expression of the gene of the DNA. As a matter of fact, even if you have for example, the tyranny of DNA, what is the meaning of the tyranny of DNA? The tyranny of the DNA can be expressed like this. Supposing you know that you have a propensity, you have an inheritance of cancer in your family. Because you're a member of that family, then chances are you have also the chance of developing. Supposing your grandmother has breast cancer, in all likelihood, you will also have your breast cancer because of the DNA. But you can escape from that tyranny by using the epigenetic factors. Meaning to say that if you modify your lifestyle, if you modify your supplementation, it is possible that those genes that you have will never be expressed because it is now redirected by the epigenetic factors. It will now be silenced. That tyranny of DNA, your destiny can be redirected. This is the meaning here. This is how to escape the tyranny of DNA. To escape the tyranny of the DNA is to operate new sets of proteins that will now express the silencing of the gene in order to change the destiny of your DNA. They use that to cure the cancer. That's why many of the cancer drugs at present are actually working as an epigenetic factor to silence the gene that is responsible for the cancer development. So that is the role of the drug. The drug there is now the epigenetic factor. So drugs are considered to be epigenetic modifiers. So you classify the drugs as genetic modifiers. To turn off the DNA responsible for the cancer is also an epigenetic factor. So epigenetics, for example, epigenetic processes generate multiple types of phenotypes from the same genotype. So starting from the same allele, starting from the same pair of gene, you can re-express that phenotype by altering what you call alternative splicing. So for example, in this one, you have your butterflies. By splicing, you can have different colors of the feathers of the butterfly. You call it the multiple phenotypes of the same genotype. That is an example of an epigenetic induction. Epigenetics, therefore, underpin the molecular, what you call orthomolecular medicine mechanisms that regulates how, when, and where the genes are expressed. So the power of epigenetics, therefore, can redirect the molecular mechanisms that underpin our body. How, the when, and the where the genes are expressed is a function of epigenetics. So this is actually, that's why epigenetic medicine is actually a supplementation medicine. So we are actually in the Forever Healthy products, we are along what you call the epigenetic medicine because our products redirects the molecular mechanisms that regulates how, that regulates when, that regulates where the genes are expressed in order to make a normal expression of the behavior of your body. The genome function, which is gene expression, is regulated by interaction between the DNA sequence, the histones, the non-histone proteins, and the transcription factors, which I said the transcription factors are the ones that are playing a role, which part of the recipe is to be transcribed, which part of the recipe to be altered, and in order to produce new proteins. So the chromatin and the nucleosomes are the key players in the genetic processes. So you see there, every package there is a histone, and that histone has a particular factor to unpack them, to modify them, to locate them, when, the where, and what type of proteins can be produced from that particular recipe. So the recipe is there, but the recipe can be altered into another expression by the power of epigenetic medicine or supplementation. So evolving the understanding of the nucleosome is actually made of 4 histones, as I told you before: histone 2A, histone 2B, histone H3, and H4. These 4 histones constitute what you call the nucleosome. So every gene is found in a nucleosome, and every nucleosome is governed by some epigenetic promoters to express that particular gene into protein. So you can just imagine, for as many as there are many genes that you have, that is also as many as equal number of promoters or equal number of epigenetic promoters in order to express the gene. Because to express the gene is to open the histone. Without the opening of the histone, the DNA cannot be translated into protein. I repeat, without the opening of the histone, there can be no expression of the gene. So now I can say that our immunogen battle is composed of enzymes and proteins that will open up the histone. That recipe could now be translated into proteins or can be translated into a particular codon that will alter the sequence of the DNA or sequence of amino acids. So histones are intimately associated with DNA and inevitably influence its function. So it is the modification of this histone by acetylation, by phosphorylation, by methylation that enables the expression of the recipe. So epigenetic medicine revolves around the what you call histone operation. To turn on or turn off the histone is a function of an epigenetic medicine. An evolving understanding of the nucleosome is actually the key role of the understanding of the epigenetic medicine. Histone tails are subject to multiple enzyme catalyzed post-translational modification. Now you can see in this diagram is an example of modification. So you have your acetyl-CoA there. It is acted upon by histone acetyltransferase or the histone deacetylation transferase. So you have there your acetylation of the lysine or methylation of lysine and arginine results to a certain expression. These kinases and phosphatases are enzymes that are able to either remove the phosphate or add the phosphate. Kinase will transfer it, the phosphatase will phosphorylate it. So we have the kinases and the phosphatases. So I can suspect that our immunogen battle has lots of kinases and lots of phosphatases that will allow the modification of the histone. So you can see there the histone is modified by acetylation and methylation. So acetylation is equated with activation, while methylation is equated with silencing of the gene. So histone tails are subject to multiple enzyme catalyzed post-translational modification in order to allow the functioning of the gene. So you have your H3 tail, you have H3 tail, H4, H2A, H2B, H3, H4. You can see there those ball-like structures. Those are amino acids in that particular protein we call histone that regulates the expression of the recipe. So the histone globular regions, the H2AK119, the HGK79, the H2B terminal, and the H2BK120 are active sites by which our modifiers were act upon. So the red ball is the acetylysine, that is an enzyme. The blue ball is the methylarginine. The green is the methylysine. The orange square is phosphoryl serine, and the red square is ubiquityl lysine. And these are the enzymes that allows the modification of the genes. So I can surmise that our immunogen battle is complete with acetylysine, methylarginine, methylysine, phosphoryl serine, and ubiquityl serine. That's why our immunogen battle is complete in this particular post-translational modifiers that would now turn on or turn off the genes. Proteins with specific binding domains recognize specific modifications and thereby influence chromatin structure and function. So multiple protein binding domains recognize specific modified amino acids and code. The one you see there like balloons, these are actually epigenetic modifiers, which I surmise that these are all present in our immunogen battle. If they are not present in our immunogen battle, our immunogen is capable of inducing our own cells to produce them. So it's either if they are not present as preformed, they can now be manufactured using our immunogen extracts. So nucleosome signaling is a beautiful. Changes in nucleosome structure through incorporation of histone variants of the actions of modifying enzymes can influence genome function. The nucleosome, as what I told you, the group of histones are called nucleosomes, a core particle that carries on its surface which regulates the gene. They have chemical signals in the form of modification of specific histone amino acids and modifications put in place and removed by families of enzymes that are present in our immunogen battle, histones carrying specific modifications by proteins by carrying defined binding domains, and these proteins will now regulate the function of that genomic region to which they are bound. So our immunogen battle is really very, very complex so that it contains some of those enzymes that will lead to nucleosome modification. So we have here the histone modification. It has a long-term and short-term effects. The short-term effects is ongoing process like transcription, replication, and repair. That is a short-term effect. But the long-term effect of this histone modification by our immunogen inductors can be inherited. That's why if you are already treated with this, what you call epigenetic medicine, it can now be heritable throughout your generation. So supposing you have your lineage of cancer before, and once you are treated with this epigenetic medicine or supplementation medicine, as I said, epigenetic medicine is supplementation medicine. So it will now cause the heritable changes throughout your generation. So one change, it will now change for the better in a long-term basis. Once protected, once corrected, it is corrected forever. That is the wonder of our immunogen supplementation. Once corrected, it is corrected forever. So nucleosome is more than just a DNA package device. It is a sophisticated signaling system that regulates the gene expression. So every nucleosome is unique. Every nucleosome holds the gene and it is expressing the genetic characteristic. It is a sophisticated signaling system that regulates gene expression. It is also inevitably that links the genomic function to environmental agents. That's why we react with our environment. We can modify it by our environment. We can be modified by our food, the food we eat. We can be modified by the supplementation we make. So this is one example of experimental agents that can alter the activities of chromatin modifying enzymes with wide-ranging effects on cell functions. So now you can see here chromatin modifying enzymes. The chromatin modifying enzymes comes from what you call environmental agents. So this is what you call the epigenetic medicine. Your supplementation is environmental agents. That environmental agents will now cause the signaling pathways to be directed in our DNA, and that is actually typified by transcription factors. And once the gene is expressed, it is transcribed, it's called a transcriptome, and it is translated into protein, and that protein will now be part of metabolic enzymes in your body that will reinforce the function of your cell. So this is now actually, and that genes could now be inherited throughout the life of that particular cell cycle. So according to Jean-Baptiste Lamarck, Lamarck is the only major figure in the history of biology whose name has become a term of abuse. Most scientists' contributions are fated to be outgrown, but very few authors have written works which 2 centuries later are still rejected with an indignation so intense that the skeptic may suspect something akin to an easy conscience. So according to Lamarck, the environment drives the heritable phenotypic changes. So this particular concept or philosophy of Lamarck still holds today. Our epigenetic medicine or supplementation medicine will drive the heritable phenotypic changes that would change the destiny of our DNA. I will repeat, our epigenetic medicine drives the heritable phenotypic changes of our DNA that would allow us to survive or that would allow us to improve for a better functional system in our body. So the paradigm is shifting. Before, they were only talking about Mendelian genetics. The Mendelian genetics is actually according to Mendel. And we came to the age of genetics where gene concept, mutation, and genotype have prevailed, that genes could evolve and genes are the very cause of diseases. But in the age of DNA, there is one of to call genetic DNA structure, the genetic code can be changed, and the genomics, and there will now be a shift. According to Darwin, the origin of species, a combination of natural selection as well as Mendelian genetics provides almost complete explanation of our diversity. So still the principle of genetic diversity underlies what you call epigenetic medicine. But DNA is just an inert coding device. As I said, DNA is just a device. Unless without epigenetic process, it cannot be interpreted and it cannot be expressed into its own recipe. But epigenetic process, therefore, can be manipulated by change in environment, lifestyle, and diet. So epigenetic medicine is actually a lifestyle medicine. Epigenetic medicine is supplementation of our diet, changing our lifestyle is epigenetic medicine. Epigenetics, therefore, is an empowering science. Epigenetic medicine, therefore, is an empowering medicine that will alter our lifestyle from here on until the next generation. So we are very lucky that our Immunogen is an epigenetic medicine because this is so empowering that would change the direction of our lives to a better life in the future. The paradigm is shifting in our age now. We are now in the age of epigenetics, therefore, is the epigenetic code, the epigenome, and the epigenetic medicine or epigenetic factors. So epigenetic medicine will free us from the tyranny of DNA. Epigenetic medicine will free us from the inherited genetic diseases. I repeat, epigenetic medicines will free us from the tyranny of inherited genetic diseases from our foreparents. Thank you very much, and thank you and good morning to everybody.